Welcome back to the subject Food Science and Technology. This is part 2 of the recording of shelf life of food. And in this section, we're going to be talking about the categories of spoilage, but then we're concentrating on chemical and microbial spoilage, since in the last video, we actually discussed the physical spoilage. So when we talk about chemical spoilage, we're usually talking about spoilage that usually or mainly affects your macromolecules. So your macromolecules and all the other food components then degrade over time. But like I already mentioned that the consequences are normally seen or observed with your macromolecules, your proteins, lipids, and carbohydrates. And the degradation of these macromolecules actually depend on the water activity, the temperature of either processing or storage, the pH, inherent pH of the product, the exposure to light or oxygen, either during processing and storage. And also, like we already mentioned that the other factors affecting this will be the presence of other micronutrients, like the presence of other vitamins and minerals. So when we talk about the minerals of importance, when we discussed the reaction lipid oxidation, your iron and copper, then accelerate the degradation of the lipids. But then the main degradation is actually observed on the lipids than on the actual minerals. So what happens is that in terms of this chemical reaction, each and every chemical reaction has its different optimum conditions. What happens is that in terms of enzyme activity, it's usually reduced at low water activity, or it's reduced at low storage temperatures of freezing and refrigeration. But then at higher temperatures, those enzymes become denatured, and therefore those chemical reaction might not take place. So what happens is when these chemical reactions that we are going to talk about just now have taken place in the food product, it usually affects mainly your sensory attributes such as your color, flavor, aroma, and texture of food. And these are actually the attributes that consumers use to actually judge food to decide whether they are to buy this or not. But one thing that, or two things that are actually overlooked in terms of your chemical spoilage or chemical degradation of your food components is that there's two consequences, whereby the first one will be when this degradation takes place, what happens is that some of the nutrients are degraded, therefore the nutritional value is now compromised or is reduced. And then second one would be some of the byproducts that are actually formed in these chemical reactions are carcinogenic. Therefore, they can cause cancer. Things like your acrylamide formation during carbohydrate processing, and also furans during processing of jarred products. And those can actually cause cancer, which are then overlooked. But the main thing that consumers are focusing on are those sensory attributes that they can actually relate with. Okay, so in terms of chemical spoilage, we mentioned that mainly it's affecting your macromolecules. So we're actually going to go through each and every macromolecule and discuss the type of chemical reaction that will be taking place, and obviously factors affecting that and so on. So in terms of the first one being the protein degradation or chemical reactions involving your protein, there's one thing that we still need to remember about proteins is that all enzymes are proteins. I remember, if you still remember that saying, all enzymes are proteins and not all proteins are enzymes. So we can say your lipase, your amylase, your PPO, your pectinase, those are enzymes and they are all proteins. But then we also have proteins like your myoglobin, your casein, and so on. They are not enzymes, but they are proteins. So in terms of protein degradation, we actually have proteins reacting with other macromolecules, one, or proteins reacting with micronutrients, and obviously the other environmental factors, whether your extrinsic or intrinsic factors affecting that. However, at the same time, then we also have your enzymes also acting on the proteins, your enzymes acting on the lipids or the carbohydrates. So those will be the type of reaction. So as I already mentioned, enzymes being complex proteins, what they do is that they catalyze some chemical reactions, and if they are present in food, if they are not denatured during heat processing, then they can be detrimental to the sensory attributes, even obviously nutritional composition of the food. So some of the different enzymes, like I already mentioned, would be your lipases, your amylase, PPO, and pectinase. Okay, so the first, in terms of your reactions, what happens is that in terms of proteins, oxidation can actually take place in the presence of proteins, whereby one of the most important protein found in meat, your myoglobin, when it's actually exposed to oxygen, it becomes oxygenated to oxymyoglobin, and then from there changing to being oxidized to metmyoglobin. And then what happens is that red pigment of your myoglobin, or your meat, your fresh meat, changes to brown, and that color becomes something that's undesirable because it shows that the meat is not fresh. So the color change from red to brown obviously becomes a hindrance in terms of the sensory attributes of the meat. And then the second one would be enzymatic activities, whereby your fruits, vegetables, and even seafood, um, your enzymatic browning can take place, and that is due to the enzyme polyphenol oxidase. So this usually happens during your processing, extraction, even harvesting. If there's any bruising, remember bruising, we mentioned that it's physical, but because of bruising, each and every reactant of enzymatic browning, being your polyphenol oxidase, your phenols, what happens is that in the presence of oxygen, all those are in different compartments within the cell. But when bruising takes place, those components come into contact, and then in the presence of oxygen, then browning takes place. So then that also affects the softening of the tissue. So it affects your seafood, your fruits and vegetables. And then the fourth one will then be what we call your non-enzymatic browning, or the Maillard reaction, which usually occurs between your protein, the amino groups of a protein, which then reacts with reducing sugars, and then it forms your what we call your melanoidins. And I mean, non-enzymatic browning is a complex reaction that just doesn't only involve your Maillard reaction. We also have what we call ascorbic acid degradation in the presence of oxygen, which then at the end of the day reacts with your sugars and then forms browning and then caramelization. So some people might not look at caramelization as a degradation product, because we do form caramels in food, and we use caramels in terms of flavoring of food. But whereby the sugar concentration is high and the temperatures are high, some sugars can caramelize, especially maybe in milk, in UHT milk. You don't want caramelized milk, so that becomes degradation in milk, but it is desired when the caramels are going to be used for flavoring. So that will be your degradation of your proteins. When it comes to your carbohydrates, the first thing that we still need to go back and mention is, we mentioned that enzymes will either react, like the enzymes are all proteins, they act on proteins or on other macromolecules. So you also get enzymes that are actually reacting on the carbohydrates, like we already mentioned amylase breaking down starch. If that's not a process that's required, and amylase actually degrade the starch into simple sugars, that is degradation, because that's not a reaction that we require. So that will be a side reaction that's taking place. Of other chemical reactions that are actually involved in carbohydrate degradation will be gelatinization and retrogradation of starch, which usually happens during freeze-thaw cycles, whereby there's moisture migration, especially in bread, whereby the moisture moves from the crumb of the bread to the crust, and then it actually dries out. So that happens not only in bread, but in baked goods, and it's known as staling. And then another thing is something that also involves carbohydrates, as we already mentioned in the previous slide, that in terms of the Maillard reaction, you have a protein reacting with a sugar. So here, same applies, that carbohydrates are actually involved in the browning reactions, whereby we have your Maillard reaction and caramelization, and also ascorbic acid degradation, which then involves the sugars. And then the third one will be your oligosaccharides and polysaccharides that are actually broken down by hydrolytic reactions, whereby your enzymes such as your pectinase actually breaks down the pectin, the methyl group separates from the galacturonic acid, and then you get softening of the fruit and vegetables, and then that's degradation because it becomes mushy. So those are then the chemical reaction that are actually involved in your carbohydrates. So this slide is just showing you the type of browning reaction. So there are two types of browning reactions that are taking place here. We have what we call your enzymatic browning. This is just a picture showing you enzymatic browning of apple juice, and then how we can actually prevent enzymatic browning by using different types of additives like your citric acid, your potassium metabisulfite ascorbic acid, and also pasteurization or heating to inactivate the enzyme. Therefore, you then reduce the rate of reaction. And this one here would be a picture that's actually showing you enzymatic browning, whereby this is an experiment we did with pulse electric field, where we show that pulse electric field enhances the production or increasing in juice yield. But one of the detrimental effect of pulse electric field on fruit like your apples will be browning, and that's actually your banana browning. So that's actually your enzymatic browning, and this is non-enzymatic browning, where we see Maillard reaction, ascorbic acid degradation taking place in your heat processed apple slices. So that is just a pictures illustrating the two types of, or we can say three types of enzymatic of browning, whereby you have enzymatic, non-enzymatic browning that's actually divided into three, which is your ascorbic acid degradation taking place, your Maillard reaction, and then lastly your caramelization that's taking place because of higher temperatures and longer storage times. Still continuing with your chemical spoilage of the third macromolecule, we actually have lipid oxidation or lipid spoilage. So the first one will be oxidative rancidity, whereby we have our lipids being either your saturated fatty acids, unsaturated, polyunsaturated, all the different types of fatty acids, whereby addition of oxygen on the hydrocarbon chain results in oxidative rancidity, and then that obviously affects the taste and the smell. It smells rancid. And then of the same type of reaction, that's actually involving a fat, but this one is hydrolytic rancidity, the second one, whereby, like we already mentioned, you actually found your proteins, which are enzymes reacting on other macromolecules. And in this instance, your lipases, they are reacting on the hydrocarbon chain, or let's say on the entire what is this glycerol and fatty acids, triglyceride. And what they do is then the lipase then breaks down the ester bond, resulting in your glycerol and free fatty acids, and that is known as hydrolytic rancidity. And that is usually common obviously in oils that are being processed, that are frying at high temperatures, frying different food products with different moisture content. But another food product where this is more prevalent will be your butter, whereby your short chain fatty acid, your butyric acids are broken down, and then you can smell that. But one thing for sure is that at times it's actually desirable. That's where you get that flavor for butter and cheese maturation. Okay, so another thing that actually then happens is that in terms of your hydrolytic rancidity, your free fatty acids that have been broken down by the lipase then become an accelerator of your oxidative rancidity. If you can see in the picture here, when your triglyceride is in the oil, your triglycerides, but what happens is that when your free fatty acids break down, what happens is that because now you actually have the hydrocarbon chain, which is then your hydrophobic, and then you've got the hydrophilic carboxylic side. So what happens in solution or in an oil, the free fatty acid would want to move, the hydrophilic portion would want to move to the surface close to where it is it's hydrophilic, and then as a result, then it is actually reducing the surface tension for oxygen, and therefore then it actually accelerates your oxidative rancidity. So hydrolytic rancidity product, then accelerating oxidative rancidity. And then what happens is that later on, especially oils that are used in frying, due to all these components being formed in here, some are aldehydes that are formed in there, other short chain carbons, what they do is that over time they polymerize, and as they polymerize, they form a viscous gummy oil. So that's that viscosity, high viscosity that you usually see for oils that have been reused and reused in terms of frying. And then later on it turns brown, turning brown because obviously of the components of food that's being fried in there. Your sugars also um, 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 being found in the oil, and also at the same time some of these aldehydes that are formed, they then react with those sugars and then they form browning. So that becomes the chemical spoilage relating to your lipids. So we've discussed all three. And then the one very much common type of spoilage being your microbial spoilage is the most common means of spoilage that can be seen. Not everybody can see that the oil is rancid. If you are used to seeing old oil, because I know there are some places where they sell used oil. So if you are used to that kind of oil that's being pre-used, you might not know the quality of unused oil, but then you will still be using oil that is actually have deteriorated. But in terms of microbial spoilage is not a good sight, especially if you can see the mold growing, you can smell the product is off, you can smell that the milk is off and it's sour. So that becomes the most common means of food spoilage. So in terms of microbial spoilage, most common microorganisms, they cause food-borne illnesses, whereby you eat the product and you might have a bit of vomiting, diarrhea, and obviously in other cases, people can even die because of that food-borne illnesses. Okay, so in terms of this microbial spoilage, its major concern is for perishable foods like your fresh fruits, your vegetable, your meat, poultry, fish, baked products, milk and juices. Unlike those other non-perishable products like your canned product that has like maybe a shelf life of 2 to 5 years. And then some of these microorganisms that are actually potential for food spoilage are your bacteria, your yeasts and mold, viruses, and parasites that will then be found in the raw material, especially your animal products. So, in terms of microbial spoilage, we already know, as we mentioned, it's one of the major. So, the growth of microorganisms can easily be prevented or slowed down because not all microorganisms can be deactivated or we can get rid of. That's why we have processes like UHT milk and pasteurization. So, in UHT milk, we get rid of everything. In pasteurization, we just get rid of the pathogenic bacteria. But spoilage microorganisms are reduced to a level that they do not multiply, provided the product is stored under the recommended conditions. Okay, so the growth of microorganisms, like I already mentioned, can be prevented by adjusting the initial microbial load. Adjusting the initial microbial load, I already mentioned pasteurization, whereby you actually reduce the pathogenic, or you kill all the pathogenic bacteria and some of the spoilage microorganisms, but there are some microorganisms that can still grow or multiply provided the product is not stored well by adjusting the temperature of storage. So if you can freeze the product or keep it under refrigeration, you can actually reduce the rate of multiplication. Reducing the water activity, because water is a medium in which all chemical reaction takes place. So if the water activity, the available water is reduced, then your microorganisms cannot multiply. We lower the pH, but like we can always mention, lowering the pH is not always a case for all microorganisms. Most microorganisms don't grow under lowered pH, but we then have your acidophiles that love acid, but all in all, lowering of pH will then result in a reduction in microbial rate of most microorganisms. We use our preservatives like the benzoic acids and the nicins and everything else that can actually reduce the microorganisms. And then obviously the last one, using proper packaging. You have to use proper packaging because in terms of packaging, the first function of packaging is actually to prevent your food product being in contact with the environment. And then you also have your special type of microorganisms, vacuum packaging. You have your modified atmospheric packaging. In that way, it then reduces or the rate of microbial spoilage. So like we already mentioned, some microorganisms simply cause food spoilage. So you look at the food and you don't want to eat it. I mean, think about when milk becomes sour. That milk becoming sour, sometimes you might not use it in your coffee because your coffee is going to go off. But I remember growing up, when the milk becomes sour, it becomes a mussy, and then we just use it for a different purpose, but we don't throw it away. So it has spoiled, but then it does not make you sick. While others cause illnesses, and depending on the severity, death can also take place. So as a result, then it means not all microbial growth is undesirable. It's undesirable if it's going to cause mold on bread, but we also want those type of mold, your Roquefort cheese and Camembert. We want those microorganisms to produce those specialized cheeses. So not all microorganisms are undesirable. Most are used in production of many fermented food products like your cheese, beer and wine, your Saccharomyces cerevisiae, that will be the yeast that's used in fermentation, soy sauce, sauerkraut, and I already mentioned the other camembert cheeses and stuff. So microorganisms, depending on which one it is, then we will see whether it's desirable or not. So this is actually a table that I took from the book Understanding and Measuring of Shelf Life of Food. It's just showing you the different types of microorganisms, the temperatures, and the food system where they will be found. So just go through it and see. Make an example of most yeast at temperatures of -5 at a water activity of 0.8, and what type of food product will you find this one growing in will be in your salted fish and stuff like that. So just go through that one. So, as we already mentioned, that microorganisms have specific requirements and conditions in which they can thrive. Okay, so some of those factors affecting your microorganism will definitely be mostly your intrinsic and extrinsic factors, like we already spoken about the nutrition requirements. We have lactose fermenters and non-lactose fermenters. So if those ones require lactose to ferment, they are not going to ferment any other sugar. They will just ferment lactose. And then presence of antimicrobial agents, when they are there, they will hinder some microorganisms, but not suitable for certain types of microorganisms. Water activity, I think it's a concept that has been exhausted, that different microorganisms have different requirements for water activity. Talk about the xerophiles that can actually grow in very low water activity, but obviously there are others that require high water activity. The pH, we already mentioned, oxidation reduction potential. The temperatures, we have your mesophile, your thermophiles, and your hyperthermophiles. So very important knowing which microorganisms can grow in a food product. We must not just assume that all microorganisms don't grow at refrigerated temperatures. I mean, think about the Pseudomonas in your polonies. And then oxygen availability, very important. When we spoke about oxygen, we talk about aerobic and anaerobic bacteria. Think about your Clostridium botulinum, which might be a big problem in terms of anaerobic canned products, especially in meat products. And then also inactivation processes. What processes are being used? Are we drying the product? Are we heating it up? Are we canning? How are those microorganisms dying in terms of that process of inactivation? So each of these factors mentioned here will definitely affect the growth. It affects whether the microorganisms does proliferate or it doesn't. Okay. So just to wrap up the categories of spoilage, this is just a table showing the major types of food spoilage, like we already mentioned, physical, chemical, and microbial. Just to wrap it up, it talks about the major mechanism for different type of food products. An example, milk. Your milk, the type of spoilage oxidation can take place, hydrolytic rancidity, and bacterial growth. So what are those critical storage variables? Definitely the oxygen if oxidation is taking place, and then the temperature, because the temperature affects both your bacteria and sometimes something that maybe we don't mention was that some of these bacteria that are growing in your product, they then produce the enzymes that then degrade obviously the macromolecules. We jump from there and then make an example of leafy vegetables. Um, Leafy vegetables, enzymatic activity, moisture loss, wilting, microbial growth. So what's so important in terms of critical storage variables for leafy vegetables? The temperature, very important, the light, oxygen, and definitely the humidity if it has to do with your wilting, the moisture loss or moisture migration. The last one, then we can talk about beer oxidation and microbial growth. So very important, oxygen, light, and temperature. So those are the major mechanisms that you need to know about.